whatever stage you're at in your education, whether you're undergrad, master's, PhD, postdoc, or whatever, or even out working and then figuring out what to be when you want to grow up, it's really worth finding out everything. Being a clone of your professor may not be what you want to do long term, or it may, and either is fine. There are advantages and disadvantages in different things you do. So I thought I'd throw a picture of my family up here. This is, I'll show a map a little bit, but this is taken a year ago last spring break in Big Bend National Park. It's in far west Texas. I'll show you on the Texas map in a few minutes where exactly it is. But what I'm going to do is just spend a few minutes going through how I got to where I'm at, some of the realities of, because a lot of times, if you're a postdoc, especially in a big med school, big research one school, okay, that's one level thing. If you get to a master's level school, you're not going to be able to walk down the hall.
Pardon me, can you please use the microphone?
Sure.
Oh, there it is.
Okay, if you're, I didn't realize this was being taped, so, okay. Part of the realities when you're at a master's level school is you have to improvise, be a little creative. There is a teaching issue, and we'll get into some of those things a bit later. But it's a lot of fun. And before I get into the formal talk, I'll just mention if you had to do something today or someday in the future that would have an impact 50 years down the line, chances are it's going to be a lot more in an interaction with somebody, maybe a bit of encouragement, maybe something you told somebody in a class, rather than discovering the next theory of relativity. I think if we're really honest with ourselves, that's a bit of a reality. So I'll just go into the formal talk. Now, just to go through some of the things that I did as a kid. I was born in 1956, so there's some real older pictures. In the upper left, I was born in Toronto, so we did have something called snow there, and I was pulling my younger brother, who was born in 1959, and I was pulling Don in a little sleigh. In 1964, just north of Toronto, that's actually at a maple sugar bush, and that's something you don't see very often these days, but it was kind of fun. I have no idea whose dog it was, but it was pretty friendly. So, and then a few years later, we moved down closer to Detroit, still on the Canadian side, and my brother and I were playing with windmills, and I used to love hanging around a local swamp, which maybe going into slime and stuff. And then as I was, hair was a little longer, not as gray, that's a picture of my family with my maternal grandmother on her 80th birthday. And then my undergraduate education was at the University of Guelph. Guelph is like analogous to a Canadian land-grant school for those of you in the U.S. It's just an hour's drive west of Toronto. And used to occasionally, in the upper left, you'd see people walking cows along their campus because it was an ag school. We'd make snow sculptures. And then the picture on the right is just a picture I'd gotten an attachment to a camera, and it's just showing a gram-negative bacteria through a light microscope. And we'd learned some good classical microbiology. Now it's very molecular, and there's a lot of excitement, but there was also some pretty good foundation back then. While I was an undergraduate, I took a number of summer jobs, and I traveled. So I'll just show you on this map of Canada different places I was. So Guelph is approximately there, just west of Toronto. And during the summer of 1976, I worked in northern Canada, a place called Yellowknife, which is up around here at 62 degrees north latitude. I'll show you some pictures in a second. I worked in a mine. That was quite an experience. And then the following summer, I got a chance to work in a research lab in Canadian Government Research Lab. And that really, I had no clue about research or even science as a potential career before that, because my experience as an undergrad was we just learned a pile of facts. You know, how much can you memorize and spew out? And that was the first experience where science actually became fun. Or some people say it's a verb rather than a noun. And so that had a big impact. And now my first academic position was at Queen's University in Canada. It's at the eastern end of Lake Ontario, just south of Ottawa and between Toronto and Montreal. But so that geographically, that'll show you where we're. In Yellowknife, this is, it was quite an interesting experience, but I was 3,000 miles away from home, first time being away from home. And so there was the trees there. This was 200 miles south of the tree line, but the trees were, the tallest ones were maybe about 8 or 9 feet tall. And the picture at the upper right, that is as dark as it got on June 21st. You're not north of the Arctic Circle, so you don't get 24-hour daylight, but they would have a midnight golf tournament there. And this was looking over the mine that I worked at. The lower left shows me, hair again longer, very dirty nose, with one of those drills they use for rocks underground. If you've ever heard a jackhammer, you know, that construction people use, imagine it in a room about half this size with that noise. So a lot of miners tend to go deaf pretty early. But there's some gorgeous scenery around there. But I figured a mine wasn't for me, and so the following summer I got a chance to work in a Canadian Government Agriculture Research Station. John Neal was the microbiologist that I worked with. That was incredible. Like, you know, as I said, you actually got a chance to do stuff. Now, even making solutions, washing glassware, all that sort of stuff, that was cool. Yeah, it may seem a bit drudgery, but, you know, when you're allowed 30 people in a class to share a beaker and then getting something like this, it is kind of cool. And then I discovered biofilms, which is, you know, sliding on rocks. And now the streams there in the mountains were fed from melting ice, so it was a little chilly, but, you know, that was fun. And then I worked for almost 4 years between my 2 degrees around Toronto. In fact, that's a skyline picture of Toronto where the next ASM will hold. It's quite a pretty city. The big tower, the CN Tower, is right near where the ASM will be next year. So I worked at a number of jobs in the food industry. To be honest, the last job I had in the food industry I got fired from. I'm serious. I'm not cut out to be a foreman at a unionized production factory. That is hard, but you do survive. You come out with your arms, your legs, most of your head. And so, but you know, a lot of stress in that. And so, you know, the thing is, don't be afraid to fail. It's okay to fail, because that way it builds up independence and stuff like that. So at the time, I lost my last job. I was living in Western Canada in Lethbridge again, and I got a chance to go to school, and it was serendipitous. I ended up talking to one of the scientists at Lethbridge research station where I was at, and he told me there's this guy named Bill Costerton. I had no clue who Bill Costerton was at the time, but I knew this guy KJ Cheng was good, so I jumped at the opportunity. And then Bill, like, the reason so many people are into biofilms is because of Bill. So the upper left is a picture on campus. That's a picture of Bill a few years ago. The lower left, if you get a chance working in a big lab, that can be cool, because one day I got heavily addicted to downhill skiing when I was in Calgary, and you know, because the mountains were an hour west of us. And one day Bill came up to me and asked me, "Bob, what are you doing on the weekend?" And I sort of had plans of going skiing. I said, "Well, nothing. What do you want?" And so he said, "Could you take these two visiting scientists, the one that's on the lower left, I'm on the left-hand side of that picture. This is up at the summit at Lake Louise. Katarina Svanborg, who is a very noted urinary tract investigator from Sweden, is in the middle, and her then postdoc, Lars Hagberg, was on the right. And so, you know, getting asked with your way paid to go skiing with somebody that's a world-class microbiologist, that was fun. And, you know, we'd ski, yak a little bit about science on the chairlift up or about whatever else. And so if you get a chance, meet people, talk to them. This conference is so great, and you'll see a ton of people going around. These are, on the lower right, are some of the people that were in my lab with me in the, there we go. This guy here, Curtis Nickel, was a urologist, and Kurt helped me get my first academic job. But we did some stuff on kidney stones together. And my PhD work, I was looking at a type of kidney stone that's caused by Proteus mirabilis, and noticed Harry Mobley, a good friend of mine at now at the University of Michigan, they've just sequenced the genome of that. Harry is a great guy. Some of you may know him. Then my postdoc advisor was Terry Beveridge, shown in the upper left here. And this was back at Guelph. We still had cattle running on the campus there, lower right. But and then two of my colleagues there, Laurie Graham. Laurie and I did our PhDs and her postdocs in the same lab, so she's my academic sister, so to speak. Laurie's here. She's at a small Canadian university, St. Francis Xavier, right now. Grant Ferris is the interdisciplinary stuff. Biology and geology is very hot right now. He's actually a full professor in a geology department. You don't think of that necessarily for microbiologists, but hey, bugs are everywhere. And so it is such a great opportunity. And so I was looking a bit at how bacterial cell surfaces, primarily capsules, interact. If you ever turn on your water and all of a sudden you get this rust coming out of your pipes, it's because you've got trace amounts of iron in the water that binds to the biofilm or the matrices of the biofilm, and it will oxidize to ferrihydrate, which is rust. So that's one example of it. The biggest thing I did during my postdoc was meet my wife. So, you know, and you know, I hope you get the impression. My family is very important to me, and it's you know, our work is great, but it's only one part of us. And so my first academic position was at Queen's University in Eastern Ontario, roughly halfway to Toronto and Montreal. It's a real pretty campus. It's on Lake Ontario. You can see in the upper right there. It's an old Canadian university, 150 years old, and it is very similar to, it's like a scaled-down version of UW-Madison, like with the lakes and stuff like that. Our two boys, Malcolm and Alistair, were born, of course, Malcolm was 3 when we left Kingston. Alistair was 18 months. Now Malcolm's just finishing a sophomore year in high school, and Alistair's an 8th grader. But and then I started at Southwest Texas. The university dropped the Southwest from its name, so we're now Texas State. And so you see Martha and I standing there with several other people. And so now, just to give you a little bit of issues where about Texas State. San Marcos is there. It's about 30 miles south of Austin, 45 north of San Antonio. And it's pretty well central part of the state. And Texas is a big place. If we drive as far west as possible to El Paso, it's a 10-hour drive. Big Bend is, talk to some of you about it, is in this part down here. But when I started there, it's a large school. It's about 20,000 when I started. It's around 27,000 now. It's in the Texas Hill Country, which is a very pretty part of the state. We have almost 1,000 majors in biology. Our department is very broad, like try to cover every aspect of biology. When I got there, I was the bacteriologist in the department. There was one other colleague who was a virologist. And so some of the issues you have to deal with, and they're challenges, but they're, if you think of them as being a real impediment to where you want to be in your career, maybe it might not be for you. But full teaching is you'll start off you'll have reduced teaching, and that should be part of when you're coming up to a faculty position, try to work things out with teaching. But full load is 2 courses a semester. Okay, for me, one of those is introductory microbiology. What happened was when I started, I had an idea of what needed to be taught, but I went and asked a bunch of friends for advice on it. And one guy who was very helpful to me was Bill Giorsi, who is now again chair at Cornell. And Bill sent me their whole syllabus and sort of abbreviated lecture notes. And I used that. I made my own notes, but I used that as a guide. And it pretty well matches the ASM recommended guide for micro. And so that helps. It gives you a roadmap of how to start things up. And then in the spring, I'll do I do that both semesters. Fall, I'll do a medical micro course. Spring, I'm developing a new course. But those are for seniors and master's students primarily. Then you'll have graduate students to supervise. Now, research, you are expected to publish. You're expected to try for grants. It's moving more to where they're wanting you to be successful. That's a bit of a transition. And again, if you're interviewing, just try and be clear exactly what's expected. A lot of places will have very flowery language. We want you to be a real good teacher, real good researcher, and real good service. Okay, the reality is, at some places, it's dollars and publications and no red flags teaching, or no convictions teaching. Felony convictions, I mean. At our place, we do try to take our teaching seriously. In fact, we've had 1 or 2 people get in trouble, as in no tenure, because they were abysmal teachers. So it varies, but just be on the level, find out what's needed. Okay, service is one thing. An ideal committee for a brand new faculty member is something like the seminar committee, because that'll give you an opportunity to bring in friends or potential collaborators or stuff like that. The committee you don't want to be on is something like space committee, where you're deciding who gets to keep their lab or not. That is a political landmine if you're not tenured. And so if you it's really more for the older faculty and chair. They really need to protect younger people because you need time. Your first year will be pretty insane because you're trying to get everything up and running. And I like to use the analogy of building up inertia. If you're trying to get a car, pushing a car, okay, give it a lot of energy at the beginning because you're going to get the odd speed bump, you're going to get the odd pothole, and your momentum will carry you through that. So that's, so I just wanted to give you that as a bit of reality. And it does get better after your first year. First year is a little insane, but it does get better. So with advantages and disadvantages, okay, in our case, we have a real big advantage of location because we're in a very pretty part of the state. And I don't know if I have any San Marcos pictures here, but we have a river that our campus owns the headwaters of. It's 71 degrees Fahrenheit year-round at Spring-fed, very clear. We have 6 endemics, like 6 unique life forms that are found nowhere else on the planet live there. And so it's like owning your own ecosystem. One favorite summer activity is tubing, where you just simply float down the river in an inner tube. And so it's real nice. Now, advantages is research expectations. Now, because you're at a master's school, it does not mean you're at academic Siberia. Okay, I am on the editorial board of Applied Environmental Micro. I'm also on the editorial board of the Geomicrobiology Journal. I've sat on a bunch of NIH panels. I'm past president of the Texas branch. I'm not blowing my own horn. All I'm saying is you're only limited by what you want. So you can really make a name for yourself. You don't have to be at the largest school or stuff like that. Quality of life. As I said, my family is very important to me. A few other things are very important to me. One day I was on sabbatical. I had a choice to go to a faculty meeting, which was optional at that time. I was tenured, so it was optional at that time. And hear somebody say, for academic misconduct, why, in spite of all the language, they were too afraid of lawsuits to do anything. I figured, I'm going to be frustrated going to that, or I have an opportunity to take my two boys fishing. And so we went fishing. We had a contest. I was a Cub Scout leader. We had a contest to see who could catch the smallest fish. And the guy that won caught a little perch about this big. So that was fun. I love the students. Sometimes they're a little overwhelming, but this was not on my radar screen when I started, but my current and former students and where they end up are probably the thing I am most proud of professionally, I'd have to say. So that's funny. Diversity of the department, we have a lot of people that are doing all sorts of biology, so something that'll be in like wildlife ecology. Never would have thought of. Most microbiologists are, work their pipettemen or whatever, and actually going out in the field collecting biofilms and then having to worry about where the next rattlesnake is or something like that, that's exciting. So and you do get the opportunity to attend meetings like this or even international meetings. The disadvantages, infrastructure is an issue. Okay, you're not going to have core facilities, or you may have to build your own. And you likely won't have a critical research mass. Internet's great. Meet people. It is so important to network. In some ways, I hate the term because it's impersonal, but just get to know people. Most of my students have had an external person on their committee just from outside the university, and I think it enhances the quality of stuff. And then there are realistically time constraints because you need blocks of time to do research. So. But you'll have different advantages, disadvantages wherever you go. Now, I'm going to just take a few minutes, show you some of the stuff that we do, and I'm just showing you little vignettes. This first bit will be an introduction. So the upper left, these are San Marcos pictures. So this is Acarina Springs or Spring Lake. It's a spring-fed lake, and the water will replace itself in this 30-ish acre lake every 8 hours on average. And so, whereas downstream, this one guy that's pretending to be a biofilm is an alum of mine, Matt Garner, who is at this meeting. Matt's at Cornell right now and is actually looking for faculty jobs. And so Matt's showing what bacteria have to do to persist in a flowing environment, which is basically attached to a surface, and you form a community like a biofilm. And then looking at a confocal image of a biofilm, this is one we actually got from the Johnson Space Center. You see, there's an analogy to the way bacteria form clumps in a nature community, to where if you were to go outside of Orlando or look at grass as it grows in a field, not on a lawn, but in a field. It forms clumps, and then there's areas where there's very little stuff. And we call the clumps microcolonies, the areas with few cells, as water channels. And then biofilms, they grow that way a lot in nature. So there's a lot of bacterial infections involve biofilms. They can be very resistant to antibiotics, and there's some good talks at this conference that'll get into that in a lot more depth. They're just, when they're growing on a surface and growing free living, they're quite different. So using, this is actually looking, it's not an aquarium picture, this is our river, so it's very clear. And you'll see some fish, and then just some water plants here. So I'm showing you an environment where there are bacteria present both planktonically and trying to attach to surfaces. And the reason I use this is because something that lives in that type of environment may figure out a way to protect itself. Maybe if we look at what happens in nature, we can find the next drug or something like that. So with that as an introduction, let me give you examples of projects that you can do with students. This is an undergraduate project. I had a student, Christy Dunn, who was very interested in fossilization, like Jurassic Park and that. One of my colleagues works with plant fossils, Gary Upchurch. And so we got thinking, if a leaf fossilizes, it's likely going to do so in a wet environment. It'll be coated with bacteria. If you spray water on a houseplant, the water beads up. The leaf surfaces are hydrophobic, sorry. And but if you put bacteria on there, it'll make the surface hydrophilic and also attract metal ions. And so the upper two pictures just show scanning EM of real fossils, and the bottom picture just shows you some sycamore leaves in a local stream. And Christy just made biofilms, dipped them in iron. They turned brown, rust brown within 5 minutes. The leaves without a biofilm stayed green for 3 months. And she got as an undergrad, got a paper, first authored paper in geology, which is that discipline's top. It's a molecular micro equivalent in geology. So that was kind of fun. And we worked with a geologist at the University of Texas. So Christy was a B student. She wasn't a straight A student. And, it's a lot of fun working with her on that. And then, now some of the stuff we've done, this will go into work I've done with some graduate students. This gets into the area called quorum signaling, where you think of bacteria as being single-celled organisms, but they can do things as a group. And they'll send signals for population density, these acylated homoserine lactones. And so with biofilms having a lot of bacteria packed tightly together, there were predictions that quorum signaling would occur in biofilms. We did a very simple experiment. Just wandered out in that little river near us, got some biofilm-coated stones, put them on I'll have to look at panel A there. It's taking me a while to get my mouse going here. And with a biosensor organism that my friend Clay Fuqua developed, we had the first experimental evidence that quorum signaling was occurring in biofilms. We ended up scooping a lot of big labs doing that. It was, now, that was, I knew people. I shared results with them ahead of time. I gave proper credit to work in progress. So, we're all trying to make a living. But you can, it doesn't always happen, but you can sometimes do some neat stuff. So, I put on the people involved. Marvin is coming to this meeting. He'll speak tomorrow. He's one of my alums. He's currently on faculty at Oklahoma and about to move to Texas. And then we've finished this up where I won't go into too much detail, but we just asked if bacterial signals could attract others onto surfaces. So the short answer is it looks like they can. We may be into a way of controlling populations. Now, this experimental detail, if people are interested, I'll talk to you privately afterwards, but I just want to give an overview of a career issue. The last thing I wanted to talk to you about was sometimes it's good to say yes when people ask you to help out with science fair projects. Okay, what happened here was a few years ago I got asked, I was in a meeting about the size of this, question, anybody here interested in helping some 8th graders out with a project that NASA might like? So I said, sure. I didn't realize these kids had an in with a company that flew commercial payloads on the shuttle. So that was cool. And so we had this, timing can help. This happened just before I went up for tenure. So we launched an experiment on the Discovery with John Glenn. And the setup was like this. On the the schematic shown with the red thing with a well, we had a small container. It's 142 microliters, the size of a raindrop. We just put bacteria in there, and we had on the top some media. It was either R2A or LB broth. And then these would be misaligned during launch and landing, and then during orbit, the astronauts would just simply turn a dial and align these, and the bacteria would try to get at the food. And then in between the bacteria and their media, we'd have a little piece of a polycarbonate filter and just see if they would form biofilms on it. It's like having a screen door when you're cooking something the flies are trying to get in from outside. And so this, in the upper left, you can see one of our astronauts, that little gray dot there. And then the lower part, this is my younger son Alistair and older son Malcolm. They were first and third grade respectively. And then some of the fifth grader friend of theirs, and then the eighth graders were hidden behind me. We had a lot of media attention. Those kids at Port Lavaca, it's on the Gulf Coast, they put out a press release. The Houston media picked it up. We had a TV and stuff like that. That type of attention, okay, ethically represent your field well. Okay, it's not you. Really, be good to the kids. It's an outreach opportunity. Those kids got paraded through town on a fire truck. And in Texas, football is a real religion. Normally you win state football title and then get paraded. So for one day, science was cool. Then it went back to normal. And then this was our microgravity-grown biofilm. And then that led, this was with some community college kids, we got a chance to launch another one. This happened just before I went up for full professor, and it was on the Columbia. And we went down and saw the launch. That was our payload. Here, I'll show you the payload on the left. And then on the right-hand side, if you look at the astronaut Kalpana Chawla, just above her head, you see something with a little sticker on it. That was our payload in the Columbia. And of course, the Columbia didn't make it. Well, this is the launch. It's an hour east of here at Cape Canaveral. And then you can see that little dot there is the shuttle about 100 miles down. And so while we were watching it, that's when the ice fell off that ultimately doomed it. And this was our payload there. It fell from 61 kilometers up and is going at Mach 18 or whatever. And it landed on a parking lot and survived. And we actually got some stuff from it, which is quite interesting. We found this bug. We just put it out. It's a microbispora. It's an actinomycete. And we think it survived the crash of the Columbia. So that was incredible. But, you know, it's a case. We had 7 people lose their lives over that. And you know, the 3 kids, students that I had, we got a ton of media interviews. I did 3 solid afternoons out of my lab, and you know, it's rare. It's rare for people to go through that, because usually in science you're very anonymous outside your own institution. But we're basically representing NASA, even though I don't work at NASA, representing your school and stuff like that. And the kids did a real good job, because you know, we're trying to grieve too, because you know, it's very moving when you think of what happened. But you know, just respecting them. So I've had people pay bills. I just wanted to mention a few bits of advice, and then I'll open it to questions after this. This is a picture of my lab from about 1.5 years ago. And you know, when you start off, you'll go into a bare room, be on your own, then you can build it up. If you can, of course money is needed, you need to get a try and get somebody to work with you as a tech or something like that. Mary Barnes, who is the lady in the back row to my right, she's wearing a medium blue top. Mary worked for me for almost 10 years, almost like having a postdoc. She'd keep the lab running, keep things help train people, and that's rare to get somebody that good. But if you can, I've given up some of my own summer salary to keep Mary on. She was that important to me, and it's get good people, treat them well, and really, I firm believer in the golden rule. So try to treat others as the way you'd like to be treated, and it works so well. And otherwise, I'll just close there and be open to questions from people or comments.